Welcome everyone to the Volunteer Water Quality Monitoring Training Part B. Just like the last training we did, all of the settings are set on mute. This training will be recorded and all of our videos are turned off. We've got additional trainers and helpers with this Stream Team program. My name is Laura Richardson, and I'll be presenting part of this, but we also have our presenter, Amy. And if we can go through and just do some intros, I think that would be helpful. All the people that are going to say hi are involved in the Stream Team program, and they assist in one way or another. So you may be in touch with them at one point within our program. So Amy, you want to say hello?
Sure, and hopefully all the notifications for this training that keep popping up on my computer will stop here in a moment. But hey everyone, Amy Meyer. I'm with the Missouri Department of Conservation, and I'm the Stream Team biologist for helping to coordinate stream teams in the what we call Missouri River Unit, and that covers the central, Kansas City, northwest, and northeast regions of the state.
Jenna, you want to say hi?
Good evening. I'm Jenna Steek. I work for the Missouri Department of Conservation. I am the Stream Team biologist for the Southeast region, and I am also part of the Volunteer Water Quality Monitoring Program.
All right. Kat, you want to say hello?
Sure. I'm Kat Lackman. I also work with the Department of Conservation. I'm the Stream Team biologist over the Ozarks. I cover basically from the southwest corner over to almost the Boot Heel. So if you need anything, give me a holler.
Randy, he handles most of our higher level data for the level 2s and level 3s. So if you continue on to the program, or if you have a specific monitoring project in mind, Randy can help out with that. So we are going to get started with today. Again, just a couple of features to point out for you all to use. If you would like to ask us a question, or some of our areas we have some response where you can provide feedback or some interactive activities, you can go ahead and put those in the chat. And we will try to follow up with you either through the presentation or through the chat box. You can also give us some presenter cues if we need to slow down, if we're speaking too quickly, or if we need to speak up. You can use this bottom right feature in your screen to give us cues while we are presenting. Okay, so for today's agenda, we're going to mostly focus on the biological monitoring. So this is identifying macroinvertebrates within your stream systems, as well as how to monitor them. We will also cover the safety and trespassing. And at the end, we'll go over watershed mapping and how to use online tools. We have a couple of resources for you all. Again, we'll have some polls or interactive activities at the end of the chapter. We'll also be taking breaks in the middle of the biological monitoring and at the end of the biological monitoring chapter. And again, you can use the chat box to ask us any questions, and we'll hang on at the end to answer any questions that you all might have as well. So we have a bunch of educational resources available to you all. Again, a lot of these are great learning tools that you can go back and reference, and those are on the Missouri Stream Team website under the Volunteer Water Quality Monitoring tab. The notebook is a good tool to reference some of the education that we provide in this presentation, and it's a great tool for following along with the presentation if you're someone who likes to read and take notes while you're listening to presentation. Also, we have the PowerPoints available on our website. And today we're going to go over a lot of macroinvertebrate information. And we have some physical tools or tools online that you guys can again use to help kind of be more familiar with the macros. So we're going to start with just some safety and trespassing overview. So we'll start with some safety features such as personal health. Again, when you are going out to your site, just make sure you're up to date on your immunizations. And also during this time period, please look up the health and safety guidelines for your local communities, as well as by the CDC. Also, please be sure that you are using proper foot protection. There's lots of jagged rocks or potentially trash in the streams. It's really easy to potentially cut yourself. So we recommend going out in the stream in rain boots, waders, or old tennis shoes versus just sandals. Also, if you've got young kids that you're taking with you, particularly if you have kind of a higher flow in your stream, make sure they have life jackets available to them. And again, try to wear sunscreen and protect yourself with bug spray while you're out there. Something you also want to be aware of is, again, protect yourself when you're going out in the stream. Pathogens are present in the water, so there's always a potential risk. Something to think about is once you're finished monitoring in the stream, just be sure to wash your hands before you eat or anything like that. So that's something good to keep in mind, is just washing your hands after you monitor. So now we're going to cover some sampling safety. Again, please just use common sense. So driving your car or heavy equipment up to the stream bank that's potentially eroding is not the most safe you can be. So just be aware again. Also, if you ever see anything alarming or suspicious at your stream, it might be a sign to leave. Do not stay and collect data. If anything seems alarming, the data is not worth it. Your safety is a priority with the program. Again, if the stream is above your knees and flowing really strong, that is too high to enter. It is really easy to fall in our streams as well. So make sure that either you have a buddy with you or you contact someone and let them know that you're going out to the stream, just in case something happens. We also have especially after some high flow events, you may find some waste that need to go through proper protocols. So if you come up and you find a drum at your site, that is something that you need to report to the emergency response hotline. Please do not touch or move the drums if you come across one. Again, you can document it, document your location, and then call the environmental emergency response hotline if you ever come across something like this. If you move or touch the drum, or if you move the drum, you become responsible for it, and that's a whole nother set of issues. So again, let the professionals handle a response for any drums that you come across at your site. You might also come across meth waste when you come to your site. This can look like everyday materials such as soda bottles or coffee filters or aluminum foil. But again, if you're seeing a lot of this or all of this together, this is a sign that potentially someone is trying to make meth in that area. So please be cognizant when you're out there. Again, if you're seeing anything like this, this could be considered something like a shake and bake kit, and it could be volatile. So don't pick it up, leave it alone, and again, document where you're at and contact your local law enforcement to help follow up with this concern. Trespassing. So please do not trespass on someone's property to get to your site. Just because you're trying to do something that's good doesn't mean you have permission to trespass. Please be cognizant of other people's properties when you're monitoring. Again, if you do trespass on someone's property and they tell you not to, or they have their property marked, there are some penalties, such as potential jail times or a fine. And again, try to be on the safe side, because even if you don't know if you cross someone's private property, you could still be doing so, and there are still potential penalties for doing so. So again, make sure you're looking up your site and doing the best you can before you go out and monitor. Some hints, again, that you're on private property are any signs that are posted, any purple paint that you might find on trees or fence posts as well. And again, maybe there's like a house or something you see like that. So that's a great opportunity, again, to ask permission, say what you are doing, and ask if it's okay to be there. We also have Stream Team ID cards, which is another tool for you guys to say, hey, I'm not lying, this is who I am, and this is what I'm doing at the stream. So here's what we're talking about with marking with purple paint to indicate private property. And again, this is marking on fence posts. So you can see here, they've got a sign posted, they have the fence post, it's painted purple. And if you're crossing a fence to get to your site, that is a definite sign that you're probably trespassing on someone's property. So again, please take heed and take note. You should not be trespassing to get to your site with our program. If you are looking to ask permission to monitor in a certain area or get to a certain property, you can contact your county assessor and use a plat map to figure out who's that property owner, so you can have that discussion with them if it's okay to monitor at your stream site. Again, here is our landowner permission form. This is a great tool to, again, explain what you are doing and who you are to help that conversation move forward for why you're asking permission. So we can't say this enough. Please take going across people's property seriously, and please be cognizant while you're at your site monitoring. If you have any questions on how to request permission, you can also ask us or contact us as well.
Hey, Laura.
All right, I'm gonna.
We did have a question about MDC land, if they should still contact law enforcement or if they should contact MDC. Do you want to answer that, or do you want me to?
You can. Do you know the answer?
Yeah. So typically we say if you're going on MDC land to let us know, and we can at least contact the area manager to let him know that you're going to be out there and kind of what you're doing, because there may be other projects going on the area, maybe in their stream or something. And if they know about it, they might be able to put you upstream or downstream, or at least that way you're not disturbing some type of science project that may be going on. So I would start with us, and then we can go from there with the area manager.
I did see the question from Sherry Carter about if we find problems on an MDC area like meth waste. Yeah, the first person you should probably call is local law enforcement, and then an agent in the county as well. So, as many people as you can contact would definitely be a good thing. So this is kind of the fun part of volunteer water quality monitoring, and my favorite thing to talk about, and that is the bugs. So here we go. We'll talk a little bit about just the introduction of biological monitoring in general. We'll go through all the identifications for all of the groups of taxa that we'll talk about that you'll be looking for in the field. And we'll take a short break before we go into the methods and kind of looking at the data that you collect and kind of how to analyze it, or at least have some speculation on what you're collecting and what it means. Okay, so If anybody wants to throw in the chat what they think that benthic means, because we're using benthic macroinvertebrates. And so what does benthic mean? And I'm seeing a couple answers come in. So yes, essentially it is the bottom of the stream. It's the rocks at the bottom of the stream, not just on top, but also underneath, and even further down as well. So benthic is the entire stream bottom. All right, macro. What does macro mean? Good, large enough to see with the naked eye. So this field protocol is based in the field so that you'll be able to identify what you find at the stream side rather than having to take a sample and go look at it under a microscope. And then of course, an invertebrate is any organism without a backbone. So these are essentially the backbone of water quality monitoring, so to speak. All right, so these macroinvertebrates are really important. We'll talk about the tolerances to pollution, but something else to keep in mind is that they are so important in that aquatic community. And they're really kind of that keystone community within the entire aquatic ecosystem. Their primary function is to convert vegetative material into animal tissue. So that puts them right in the middle of the food chain. So without these macroinvertebrates, you're probably not going to see fish in the streams that you monitor. All right, so the great thing about monitoring for macroinvertebrates, they really tell us a lot because they're relatively not mobile. Now, in the event of some kind of toxic event where something toxic gets dumped into a stream, sometimes a fish can at least swim up or downstream to escape it. But these macroinvertebrates really cannot. They are very limited in their mobility and how they're able to move along the stream. They're very easy to collect, and most of the equipment that we provide is pretty inexpensive. So we are happy to provide the collection resources for you guys, which is a whole part of the program. And they do have those different pollution tolerances, and we'll go into a little more detail about that. You can monitor for them, we say continuously, but we don't mean all the time. Particularly, we only want you to actually monitor for macroinvertebrates twice a year, in the spring and in the fall, because in order to collect that information, you are actually disturbing the habitat. So we don't want to be out there all the time collecting invertebrates because that really does disturb their livelihood and their ability to grow and continue through their life cycle. And then, maybe contrary to popular belief, we do say they are easy to identify, and that's because we're taking them to a higher taxonomic level, to the order level. And of course, there's no chemicals needed in order to do biological monitoring. It's all physical. So, regarding taxonomic classification, for the most part, we are actually looking at mostly that order level. However, there will be some organisms that will go a little bit into some lower taxonomic levels. And what I mean by lower is more specific into that genus level. But for the most part, we're typically taking them to the level of order, which we can easily do in the field. And we do have three main categories of pollution tolerance, and that is pollution sensitive, pollution somewhat tolerant, and pollution tolerant. So that kind of simplifies things a little bit and breaks it down to only three categories. One of the resources that you would have received in your registration, or if Jonah wants to drop the link from our website into the chat, or Kat, also, thank you. This is a really great resource to have in the field, and we definitely do recommend laminating it. But we have plenty of those in stock, so if your blue bug card gets wet and mushy, we can easily replace that for you. But this is a really great resource because it breaks down those three different categories of Group 1 taxa, which are your sensitive organisms, Group 2 taxa, which are your somewhat tolerant organisms, And the great thing is, too, you have a size reference. So if you're not sure what you've got in front of you, you can kind of take a look in real time on that blue bug card to see, does the size match to what you've got in your hand. And so the full bar is the maximum size, and it is pretty true to size on the actual publication if you're looking at an 8.5 by 11 blue bug card. And then the black part of that size bar would be the minimum size. So you'll notice some things can get pretty big, especially this guy here. He can get pretty large, but early on in its development, you might find one that's a little bit smaller. And then if you look at this guy, this riffle beetle, that's the maximum size. That's pretty much as big as they get, and that is pretty true to size. So this is a really helpful document for you to take to the field. So our group 2 taxa, which are somewhat tolerant, continue on to the back side. And then our group 3 taxa are our most tolerant organisms. And I'm going to back up just a second just to see if you guys can notice kind of how the form and shape of these organisms change as they become more into that tolerant category. And you might notice that our group 3 taxa are pretty simple. They don't have a lot of appendages, typically don't have legs. They most definitely do not have gills. So these are the ones that are most tolerant to not just any type of pollution, but also oxygen as well. So they might be actually getting their oxygen from the air rather than from the water. Okay, so we'll go right into the identification. Again, really great resource. Definitely use that blue bug card. And we'll go right into the anatomy of a mayfly, our very first one on the list. So a mayfly nymph, which is the immature version of a mayfly, and I'll use the word nymph and I'll also use the word larva. They're somewhat interchangeable because they're both in that immature stage. However, a nymph is going to be more descriptive of that group that has that incomplete metamorphosis, which means that they emerge directly into a subadult or adult stage. A larva, on the other hand, is going to have that complete metamorphosis, which includes that pupa stage in between the immature and the adult. But mayfly is a nymph, and in that immature stage, they still have a head, a thorax, and an abdomen, the 3 main insect parts that pretty much all insects have. They do also have legs, so 6 legs. Being in that immature stage of that incomplete metamorphosis, they do actually grow wing pads as they grow in the water. And excuse me, typically it's going to take about a year or so for a mayfly to fully develop before it becomes an adult. And then it'll live for about a day or 2 as an adult. So this is really where all of that energy is going into, is in that immature stage. And as they grow and molt several times throughout the year, they will continue to grow those wing pads, which will fully emerge when they actually split open and emerge into the air. Now, the main character that we want you to remember about mayflies is that they have gills along the sides of the abdomen. Okay. Now, the first thing you're going to notice when you see a mayfly are those 3 tails, but we'll actually see some other organisms that may have 3 tails as well. So that'll be the first thing you notice, but we really want you to look for those gills along the sides of the abdomen. So, for example, and they do come in all different shapes and sizes, and those gills can look a lot different depending on the taxa group of mayflies within that order. But you'll see those gills, and they'll move those gills. They'll move them up and down, side to side, to try to get that oxygen across their body surface. And again, they do have 6 full legs, segmented legs, and mayflies have 1 claw at the end of each leg. And typically 3 tails, but again, that can be tricky because sometimes in the sampling process, you might break off those tails, or some mayflies do actually only have 2 tails. So again, it'll be the first thing that you notice, but we really want you to see those gills. and that's going to help solidify the identification of the mayfly. And again, they do come in all different shapes and sizes. So these particular gills are a little more plate-like, and they may not move them as readily as you might see in some other groups, but definitely look for those gills. Okay, any questions about mayflies? All right, we'll move on to the stonefly. So again, it is an aquatic insect, and it is a nymph with that incomplete metamorphosis, and still has that head, thorax, and abdomen. However, if it does have gills, and not all of them do, they're only going to be on the thorax, and they're going to be right at that point of attachment to each leg. But the first thing that you see for sure is going to be those two tails at the end of the abdomen, and then yes, look for those hairy armpits. And one of the cool things that you'll notice about stoneflies is when you have them collected in your ice cube tray, they may start doing push-ups. They'll actually do push-ups with all 6 of their legs up and down to get that oxygen over those gills. So hairy armpits and push-ups. It's really fun to see these behaviors when you encounter them. But no gills along the sides of the abdomen because that is restricted to the mayfly. But then those 2 tails are going to be pretty prominent. And for me, I know I have a stonefly immediately when I kind of see 2 tails on each end of the organism. So they have those really long antennae, typically, and also those 2 long tails. So you're looking at 2 points on each end of the body, but no gills along the sides of the abdomen. And they can be quite colorful and decorated. So this is your typical golden type stonefly, and you can really see here those little hairy armpits, those gills right along the edge of the thorax where those legs are attached, but not on the abdomen. And again, like mayflies, they do come in all different shapes and sizes, and this particular kind is called a giant stonefly, and they do get pretty big. They can get up to about 3 or 4 inches long, and they're living in the stream in this immature stage for up to 3 or 4 years as well. So again, all the energy is going into the development of that nymph, and they have a very short lifespan as an adult. And for stoneflies, it might be up to a couple weeks. And that will vary among the different groups. And they usually typically don't go very far from where they emerge. So that is a quarter for size reference for the giant stonefly. Pretty impressive creatures and really common in our Ozark streams. You can really see those wing pads too. And you can really distinctly see both sets of wing pads there on the thorax. Okay, moving on to the caddisfly. Now the caddisfly does have a complete metamorphosis, so it has a pupa stage in between that immature and the adult. They do still have the head, thorax, and abdomen. However, and also the 6 legs, but they also have what's called prolegs. So these are not true walking legs. Essentially, they are there to help attach them to the substrate so that they don't drift downstream when the water rises during a flood situation. They can anchor themselves to their substrate. And so there are hooks at the end of those prolegs as well to help anchor them. And you might notice on this particular specimen, it does have gills along the abdomen, but along the ventral or the underneath side of the abdomen and not along the side. So hopefully, based on this type of body shape, you won't confuse these with a mayfly, because the mayfly gills are always along the side of the abdomen. And we do like to say that they're a little bit crunchy on top and cream-filled on the bottom, and that is an old Lion King reference of all of the crunchy and cream-filled critters across the land. So these are a really good snack for fish, really great for fly fishers. If you are out there and kind of looking to see what's emerging when you're ready to catch some trout, caddisflies are one of the tastiest treats out there. And a really great thing about caddisflies, which make them really interesting, is that they produce soap, much like a moth or a butterfly does to build its chrysalis or its cocoon. And so their particular silk, they actually produce it from just under their head, and they will take bits and pieces of their environment and build a case around themselves. Certain groups anyway. Others, like this big guy here, he will actually build a net in the actual flow of the water to help catch his food. And so they'll kind of pick apart what they find that flows to them and then spend most of their energy when they're not feeding to repair that net. But then they will take bits and pieces of rocks and leaves and sticks, woody material, to build their cocoon in order to pupate before they emerge as an adult. So, cream filled. I know it's about dinner time, right? So here's just another example, and this is a net spinning caddisfly. So you do see those gills, but they are on the ventral or the bottom side of the abdomen. And you can see those really long prolegs. So he can hook himself to his substrate pretty easily. And again, they do have that crunchy head and thorax, 6 full legs, crunchy and cream filled. And again, another example where you do see those hard plates behind the head. Oh, let me go back for just a second. But yeah, those prolegs are definitely very prominent. Again, they don't walk with these legs, but you can see the hook at the end of each proleg there to help anchor them in a flowing environment. And this is kind of an example of what those nets look like. They are full of silk. They're kind of messy, but he's protecting himself while being able to catch food in the meantime. And then the ones that take the silk and those bits and pieces of rocks can build a little motorhome for themselves to protect themselves from fish. So this is a really interesting and advanced adaptation for protection. And some species, they're very particular about the type of case that they make. And they will chew bits and pieces of those leaves into perfect little squares, and then build themselves a log cabin that might be perfectly square. And you can actually identify these to family and even sometimes to species based solely on the type of case that they create. So again, fascinating, very industrious creatures. And this is all just bits and pieces of little leaves that they chewed very intricately into little squares. Now, I wish I had thought about this before, but I could be rich. There are artists out there that actually cultivate caddisflies to create jewelry because they will take bits and pieces of their environment. If you introduce certain things, just like bits and pieces of gold, jewels, pearls, they will create their own little piece of art to live in, and then close themselves into it with that silk and pupate, emerge unharmed, and then leave behind this beautiful piece of jewelry with that really strong silk. It's really strong to be able to withhold its hold underwater. And there could be some medical implications with that as well. I've read a little bit about internal suturing using caddisfly silk. And I haven't heard much of an update lately on that, but I thought that was a really great idea because they can be cultivated and kept unharmed. All right, moving on to the riffle beetle. So we have 2 types of beetles that we'd like you to know, 2 that are in the sensitive category, and the riffle beetle is one of them. And we do count the larvae and the adults together, so we combine those for your count. But they are very, very tiny. As you can actually see the fingerprints on that photo of how small they are. And again, going back to that blue bug card is going to be a good reference in order to identify if you've got a riffle beetle. What's interesting about these is that the larvae are fully aquatic. The pupae, because they are a true beetle, are actually on land or on the edge of a stream, and then they return back to the water as an adult. So a little bit closer look, and you might remember that caddisfly is crunchy and cream filled. However, the riffle beetle larva is crunchy all the way through. And again, very small, not much larger than a quarter of an inch tops. And the adults are really tiny as well. But they're very comfortable in the water, and you know you've got a riffle beetle if it just hangs out on the bottom of your sorting tray, and it will try to cling to anything else it might be close to. They're in that riffle habitat. All right, the other beetle that we want you to know in the sensitive category is the water penny beetle. And only the larvae are what we're going to find in a riffle habitat. The adults are fully terrestrial. But if you kind of look, it kind of is a little bit like the shape of a penny and pretty close to the color, like a lot of copper color in them, but very primitive looking. But if you take a look underneath, they do have all of the characters of an insect. They have a full head, they have 6 legs, an abdomen, and often gills underneath as well. And you'll be able to notice these, they can really camouflage themselves very well. But if you do happen to get them in your sorting tray, they will very slowly glide across the stream bottom or across your sorting tray bottom. And they're using those little legs, but they're very good at hiding themselves. Okay, next we have a gilled snail. And if you've noticed, one of the main characters that we have in common through all of these sensitive organisms is they have gills. And so they're obtaining that oxygen directly from the water. And that's really important. And of course, we talked about riffles and site selection and discharge, and how important that habitat is for the macroinvertebrates because of that introduction to oxygen into the environment. But if you're looking at a gilled snail, and let me see, kind of a mirror image, so I don't know if you're seeing it right, but if you hold up the snail and the opening is to the right, then you have a right-handed snail, and that's typically going to be a gilled snail that's getting the oxygen from the water. And these guys too also have a little operculum, or what I like to call a little trap door, and they can close that in when they're stressed out. So they're very hard-shelled, very sturdy, and then that little trap door they can close, and of course their opening is to the right. You can kind of see that there. All right, so in the chat, tell me how many snails you see in this photo. So I'm curious. They're easy to camouflage as well. They can easily hide themselves. Okay, see 8. Any other guesses? Ooh, 10. 11 or 12, yeah? There's a lot there. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. There may even be more in there that we missed. So they are easily camouflaged in their environment. Okay, so one of the resources that you got also before joining us, and we can put that in the chat as well, is the Key to River Life, which is a visual key in order to take 2 options and be able to decide which one fits best for the specimen that you have in front of you. So we'll kind of run through this pretty quickly. If we were in the classroom, it would be a little bit different, but I'll try to run through this quickly. So does it have a shell or no shell? And when you think of a shell, you think a snail, a clam, perhaps. So it does not have a shell. Does it have legs? Okay, yes, it has legs. And now you can move on to the next couplet on how many legs. And the legs on an aquatic insect are always going to be along the thorax. So these are the legs. So it does have 6 legs. Does it have wings or wing pads? And no, it doesn't. That's something that we saw on the mayfly and the stonefly, but not on this particular creature. Does it have tails? And what we mean by tails are those long hair-like tails that we saw on the mayfly and stonefly. And no, it does not have distinct actual tails. So if we take a look at that key to river life, and so we went through the whole shell or no shell, so no shell, legs, 6 legs, wings, no wings. So we go all the way down here, no obvious tails. What do you guys think we have here? It's not a caddisfly. And sometimes it takes a little time to really get used to using a key like this. And maybe the images could be a little bit better. Nope, it's not a dragonfly. Nope, it's not a beetle. You guys are great and you're paying attention, which is awesome. So, okay, this particular thing says 1 or 2 tails. Now, if you go back and you look, the end does have, oops, whoops, there we go. I just gave it away. It is a damselfly larva. So sometimes you have to really have that specimen next to the key. And using a text-based dichotomous key sometimes might be a little bit better, as far as kind of confirming what you have. So use different resources and we can provide those for you as well. The damselfly, definitely, you can see that better in the blue bug card. So definitely use that resource as well. So damselflies are the coolest thing. And they do get very large and up to 4 inches long and often up to 4 years in that larva stage. They do have that complete metamorphosis. But you'll really notice those big mandibles. Those mandibles are very chompy, and they're not afraid of you either. So always be careful when handling pelgrimites or damselfly larvae, because as you can see, they can inflict some damage. I've never had one break the skin, but they'll try. So one thing to be careful about if you're walking around in a stream, we definitely don't want you to do it barefoot. And if you're lifting rocks, just be careful. Now, they do also have those gill tufts underneath, and that, again, is one of those characteristics that we're seeing in common with all of the other sensitive taxa. And then they also have these long filaments along the sides of the abdomen. And typically, those are more sensory in nature. But these are definitely predators. Okay, moving on to somewhat pollution tolerant. That is part of the group 2 taxa that starts on the first page of your blue bug card and continues onto the back page. So we have a crayfish, and of course, if it walks like a crayfish and swims like a crayfish, tastes like a crayfish, we know it's a crayfish or a crawdad, or a mud bug, or a ditch cricket. So they do have lots of fun nicknames. And these are really important, even though they're somewhat tolerant to pollution. As far as the ecosystem goes, in that community, they're one of those keystone organisms because they eat a little bit of everything and everything eats them. So very important in your stream, and we hope that you'll be able to find these in the streams that you monitor. Okay, and also crayfish are a type of aquatic crustacean. And another type of crustacean is the sow bug, the aquatic sow bug or aquatic pill bug. So they're very closely related to the roly-polies that we might see in our basements or under rocks around our house. But instead of rolling up into a ball, they are actually flattened from top to bottom. So dorsally-ventrally flattened is the way that we describe that. And you'll see all those legs. So 10 pairs of legs. It's pretty distinct. And they really love that organic material. So in streams where you have a lot of nutrients, input into the stream, these are going to be pretty abundant. All right, and we have another crustacean, also closely related. And if you guys want to throw in the chat, what does this look like to you? Any guesses? All right, yes. Yes, it definitely looks like a shrimp, doesn't it? So they are very closely related to freshwater shrimp, which we do have in Missouri. However, actual freshwater shrimp are living in the top of the water column or along the edges of the stream, and you won't find them as readily on the stream bottom. So remember that we are looking at the organisms that live in the benthic zone or on the stream bottom. And these guys do. They really love crawling on those rocks. They're also called side swimmers, and they swim on their sides. And they're really fast too. Once you get them in your net, trying to catch them sometimes can be a challenge because they really do scoot pretty quickly on their sides. And again, they do have 10 pairs of legs, but they also have 2 pairs of antennae. So pretty distinct, and they usually kind of have that cream or sometimes even an orange or pinkish color to them. So another example, you can kind of see he's missing 1 antennae there, but they do have 2 pairs of antennae. All right, next up is the alderfly larva, which is closely related to the damselfly or the hellgrammite. This is kind of where we're getting into that taxonomy, into a lower level of being able to identify them to closer to species level. So we're actually getting down to family and genus with the Megaloptera. And of course, they're called Megaloptera because they're large. And so the damselfly is an easy identification. Those gills underneath the abdomen are going to be pretty distinct, but the alderfly does not have gills underneath its abdomen. It does still have strong mandibles, so it is a predator. No gills, but they do have those filaments along the sides of the abdomen. And then their tail, instead of kind of like those 2 little hook-like things that we saw in the damselfly, this one has a single tail. It almost looks like 2 pieces that merge into one that kind of looks like the letter A. So it's a single long tail, but it looks kind of like 2 that come together. And so think A for alderfly. Most of these are going to be the most common in our Ozark streams and typically just above a riffle habitat. They're not as common inside the riffle itself, but maybe further downstream or a little bit upstream. And just to confuse you all a little bit more, here is the other megalopteran that we would like you to identify if you see them, and it's the fish fly. And very closely, again, related to the damselfly, and you really see those mandibles. But again, no gills under those lateral filaments. And then they do have 2 prolegs at the end of the abdomen. You'll definitely notice those mandibles. So they got chompers. So also handle with care. If you get a chance to look close enough, and we'll provide you with magnifiers too if you're not sure what you've got when you're collecting your specimens. A fish fly is also actually going to have breathing tubes on their 9th abdominal segment. Now, this is going to be hard to see in this photo, but that's something to look for, and that's actually their ability to breathe in the water. But no gills underneath, those long lateral filaments, and that can distinguish it from the damselfly. And typically, you know, the damselfly kind of had that tattooed pattern on its head and body. They're very decorated. The fish fly is going to be a little more bland and dark. A lot of times the head and the thorax are actually black or a dark brown. Okay, moving on to the damselfly, and again, we are still in the somewhat tolerant category. All right, so what do we see? We see 3 tails, don't we? What else had 3 tails? You guys want to drop that in the chat? Right, so the mayfly also had 3 tails, so it's the first thing you notice about an organism, isn't it? Are the tails. However, for a damselfly, these tails are actual gills. So they are obtaining their oxygen from these oar-like tails. And again, you'll see that they do not have gills along the sides of the abdomen. So that's going to be your major distinction between a damselfly nymph and a mayfly nymph. And a damselfly is closely related to a dragonfly. And you may recognize them as adults. They're very delicate. Oftentimes they're colored blue. They may have black wings or clear wings, but they're very delicately kind of, flighty, flitting around the banks of a stream. And they are very delicate, but they are also fierce predators. What's really neat about them is they have this movable mouth part underneath that they can actually control. So their lower lip or their labium actually can extend to grab their food. So they're feeding on the mayflies, the stoneflies, the scuds, anything that comes to them, and they're good at really just sitting and waiting for prey to come to them. Again, aura-shaped tails that are actual gills. And a lot of times they do have pretty long, spindly legs, but you'll notice those 3 tails, definitely. But look for gills, and if you don't see gills along the sides of the abdomen, you've probably got a damselfly. And they are in that incomplete metamorphosis group where they do grow wing pads as they continue to grow and molt in the aquatic environment in that immature stage. And usually their life cycle is about a year. All right, and then their bigger cousin, the dragonfly, one of the most beneficial insects for humans. And so anything that we can do in our aquatic environments, and they're pretty much found in every aquatic environment, streams, ponds, wetlands, lakes, anywhere where there's water, you are hopefully going to find dragonflies. So very important for human health because of their voracious appetite for mosquitoes. And not just as adults flying in the air, but also as nymphs in the water. And here you can actually see that little mask-like lower lip that they can extend to help grab their food. And they're really good at camouflage as well. But you'll notice they have a much fatter body. They're pretty stout and large eyes, so they are true sight predators. And definitely spindly legs. And there's that mask-like lower lip. And it's so funny. I love it. It's like these teeth together, and they can just, it's just really cool to see that in action. So look for YouTube videos of dragonflies in action, and it's really awesome. But yes, definitely they're a lot more stout with a wider abdomen and big eyes. So here's a couple more examples, and they do get pretty big. The dragonfly life cycle can be anywhere from maybe 6 to 8 months up to a couple, 3 years or so. So they're variable depending on the habitat that they are part of, and then the type of food that they have available to them. Oops. So here's just a few more examples of the type of dragonflies you may encounter. And some do have much longer spindly legs than others, and some have kind of longer, more cylindrical bodies. But these are what you will encounter. And look for those big eyes. Look for those wing pads. And they do actually have an internal gill inside the end of their abdomen, which is really fun if you collect some dragonflies and take them to show the kids or to, an event or just to observe. They aren't really good swimmers, and really their legs are kind of chunky too, so they're not really good at moving. But what they can do is they can actually shoot water through their body, through that internal gill, anal gill, and actually shoot water out of their rear end. And so that will actually jet them across to get them where they want to go. That's a good one for the kiddos, and a good example of what we call a butt breather. All right, next up is a water snipe fly, and this is a true fly in the Diptera order. Not as common in Missouri, but we do find them in Ozark streams, and they are, again, in that somewhat tolerant category. Being a true fly, the dipterans do not have 6 walking legs. They're all kind of, which is funny that they're considered to be more advanced, but they do kind of all have that kind of maggot-like structure to them. So this is actually the head end here. And then on the rear end, they have 2 feathery horns. You can really see those body segments, and they're almost caterpillar-like. And each one has these prolegs with little crochets or hooks inside of them. And again, this is going to be something that's going to help them anchor to that stream bottom and that benthic habitat. Those 2 feathery horns are going to be your primary character in order to identify these guys. Again, not real common, but you'll find them in Ozark streams. If anybody is monitoring, perhaps in the Gasconade, Ruby Dew, Big Piney, Black River, Current River, any of those scenic Ozark streams, you might encounter these. And they can actually get pretty big, up to about 1/2 inch or so. And that creamy white color is pretty standard across these guys as well. Now, what you will encounter more than anything everywhere in the state is a horsefly larva. And actually, they are not part of our protocol, but if you get them in your stream sample, we do have a comment section on the data sheet where you can say, we got a horsefly. But the main things that you're going to see, the differences between these two, of course, again, with the water snipe fly, those feathery horns, but with a horsefly, they're actually pointed on both ends. Okay. So I believe this is the head. It is retractable, but it's not a developed head, so to speak. So, but along the entire body, they have what we call creeping welts. Actually, this may be the head end. I'm not positive. I'd have to see it in motion to be sure. But the horsefly is going to be something you're going to find everywhere, all parts of the state. And they do look very similar to the water snipe fly, but they are pointed on both ends and do not have those feathery horns on the end. Okay, up next is the crane fly larva. I just love these because they're just super squishy. You can almost put your finger down in the middle of one and just watch all the little innards of these guys kind of move around. And they get really big. They do only have about a year-long life cycle, but again, they're spending all of their energy in that larval form. It is a true fly and does have the true complete metamorphosis. The pupae of these guys is pretty fascinating, and you can probably catch that. The pupae, they typically emerge most often in late spring, early to late spring, so like April is probably your prime month for crane flies. And you might know them by the adults looking like giant mosquitoes. So that giant mosquito with the really long spindly legs that gets stuck in your screen door is a crane fly. But no worries, they are completely harmless. And they do not have functioning mouth parts as adults, so they cannot bite you. They are completely harmless. But as that larva, they are super squishy, and then you'll notice at the end of their abdomen, this is the end, and that is the head, they do have these long finger-like projections. So that's kind of to help protect their breathing spiracles here at the end of the abdomen. Awesome fish food. So if you collect these and you have an aquarium at home, your fish will go crazy over them. Okay, so just to see if you were paying attention, what were the 2 beetles that we wanted you to know in the sensitive category? Go ahead and drop that in the chat. If you remember, it already feels like it's been a while. I'll give you guys a second. So we had 2 beetles. Okay, yep, the water penny beetle. What was the other one? Yes, the riffle beetle. Okay, good. You guys are paying attention. So, okay, we've got about 1,000 species of aquatic beetles here in Missouri, well, North America in general. There's many beetles that still have yet to be described. So we want you to know the 2, the riffle beetle and the water penny. The other 998 fall under what we call other beetle larvae. And so this is kind of a tough one. It's tricky. And so having that blue bug card, that key to river life, and then all the other resources for being able to compare photos or pictures of these organisms is going to help you to kind of narrow it down. And it really is a process of elimination because there are so many different types. But the other beetle larvae are in that somewhat tolerant category. They may have characteristics that are just like everything that we just talked about. So this particular guy, he's got big mandibles, like our Megaloptera. So like the damselfly and the alderfly and the fish fly. However, there are no lateral filaments on this guy particularly, although some of them do have lateral filaments. And this guy actually has 2 tails, kind of like the stonefly, right? So being able to kind of get that search image for identifying those main characters and then seeing which ones are present and which ones are not will kind of help you to identify those other beetle larvae. And these are going to be really prominent in later spring, so late April, May, even into summer as well, and they do get pretty big as well. So just use the process of elimination. Okay, freshwater mussels. These are really important organisms. They may be somewhat tolerant to pollution, but because they are filter feeders, they are taking in everything that the water will provide to them, and they can handle it. However, they are extremely imperiled because of the way that we have altered their habitats, especially in our larger rivers with channelization or straightening the stream, building dams. That kind of thing. Any kind of physical habitat changes we make to streams can negatively impact freshwater mussels. So if you do get freshwater mussels in your sample, just make sure that you put them back right away to let them go back to doing their work of filtering water. These are very long-lived creatures. Some of them can live up to almost 100 years. So we really want to be as gentle and delicate with these guys as possible. However, we do have some invasive or non-native mussels here in Missouri, and you can save those as specimens for your reference collection, particularly Corbicula or the Asian clam. And the main difference between a native mussel and an Asian clam is that the Asian clam is going to be a little more round, maybe almost kind of teardrop shaped, with very strong ridges that you can kind of run your fingernail, your thumbnail over them. You can really feel those strong ridges. And that's going to be your Asian clam. And we definitely, you're fine to collect those, take them out of the system. They are non-native, but they still do filter water, so they do still serve a purpose, and they've been naturalized here for well over 100 years. And they do live simultaneously with our native mussels as well. But if you get one of these native mussels that's a little more oblong, kind of irregularly shaped, might have little bumps on it and irregular ridges, those native mussels, we want to take care of them and put them home. Okay, pollution tolerant. So we're down to the last group of taxa, group 3 taxa, the most tolerant. So worms are going to be something you're going to find a lot of. And again, I don't know if I really emphasized this earlier, but it's okay to find tolerant organisms in your stream because we want you to count everything. It all matters. Everything has a place, and the most diversity that you have is going to be the most beneficial for your stream. So aquatic earthworms, of course, are closely related to the earthworms that we might see on the sidewalk after a good rain, but they are fully aquatic and segmented. And then horsehair worms are not segmented, and often they can be very long, which kind of lends their name as a horsehair worm because it looks like a horsehair. And these are really fun, and for the sake of time, I won't go into much detail, but they do have a very interesting life cycle. And if we want to talk about that later, feel free to email me, or if we have time, we'll talk about that a little bit later, but they are unsegmented. They're harmless to humans, so something I wanted to emphasize as well. All right, so next up we have the midge fly larvae. And so we also know these as chironomids. They are the bottom of the food chain. Everything eats them, so they're still very important to have in your stream. And actually, the groups within this order can be extremely diverse, so they are still beneficial. But they're pretty small, and you might find them trying to crawl through your net because they're small enough they can almost get through that mesh, and sometimes they're successful. So grab them when you see them as quickly as you can. They are a true fly larva, and they do actually have a head capsule, unlike some of the other flies that we were just talking about in the somewhat tolerant category. So true head capsule with eyes, but they also will have these tiny little prolegs. So not true legs, like we saw with the caddisflies. None of the dipterans have true walking legs, but they may have these little nubs right behind the head and right at the end of the abdomen. And it's just a single proleg there. Very important, but they are tolerant to pollution. So look for that head capsule, and the size is going to be telling on this. They're going to be much smaller than your typical caddisfly, but again, look for those walking legs. If you don't see them, but you might see that little nub, then you've probably got a midge. Now, some of them have a red hue to them, and we call them bloodworms, and that's actually because they produce hemoglobin. And if you have taken biology, you might be familiar that hemoglobin is the primary component of making our blood red. And so they are able to utilize that particular component for them as well. The bloodworms are actually probably the most tolerant, and I've collected samples, put them in formalin or formaldehyde and alcohol, and a few days later got some guys wiggling underneath my microscope. So they can put up with a lot of anoxic or oxygen depleted types of environments. But again, they're still important. Okay, here we have a black fly larvae. Again, another true fly in that Diptera order. And black flies are really kind of fun and interesting. You'll know you have a black fly when you put one in your sorting tray. You won't know what it is necessarily on your forceps, but it'll immediately stick its little fat butt along the sides of your ice cube tray. And so it has these little suction cups at the end of their abdomen, and they really want to stick. They just want to stick to anywhere that they can. And they really love that riffle habitat, but they can handle a lot of pollution. But they're kind of fat, lazy butts too, because they're just gonna stick their butt to whatever they can, and then they're gonna take those little fans at the top of their head and to collect food from. So they don't put a whole lot of effort into their feeding. You notice that bowling pin kind of shape to them. So their little heads are little filter feeding fans. And yes, they have those fat butts. So here's kind of an example. And they can conglomerate into large numbers. Really here in Missouri, thankfully, they're not that populous. I mean, you'll find them for sure, but I've flipped over rocks up in the Pacific Northwest and just seen thousands of them all smushed together under a rock. And adult black flies do bite, and they can be quite problematic, and they are definitely a lot more prominent in the northern part of our continent. But yes, look for that fat butt, and it's going to stick itself to whatever it can. And then look for those fan-like structures. Okay, so a leech is also in that tolerant category, but I have to give a little love to the leeches. Most people are very grossed out by them, and I understand. They are bloodsuckers. Typically only temporarily blood-sucking, but when they are feeding on the blood of us or fish, turtles, other organisms, that's typically in the summertime when their other food sources have emerged. So they are typically predators. They do prefer to munch on black flies, midges, even maybe mayflies and stoneflies if they can get a hold of them. But in those times, especially in the summertime, when most of that food source has moved into the atmosphere or into the air, they're going to look for whatever they can to feed on. Now, again, I have to give them a little bit of love because even though they are creepy crawly and they make you bleed, they are not disease transmitters. So they're actually beneficial, in fact, in the medical field, because they have that anticoagulant, and when they bite you, you don't feel it. And so they've been used for centuries, maybe even 1,000 years or more, to help reattach fingers or toes, parts of your body, because they can get that blood flowing without you feeling it. And again, they don't have the disease transmission issues that mosquitoes, ticks, and some of those other blood-feeding organisms have. So just want to throw a little bit of appreciation out for the leeches. And they can be quite pretty as well. They do have suckers on both ends. And typically that more narrow end is actually the mouth, but they will have a suction part on the rear end as well. Okay, and so you'll definitely encounter leeches all over the state. Contrary to popular belief, they do enjoy flowing water as well as stagnant water, so you'll find them pretty much everywhere. And when you see them, they will stretch their bodies out and stick one end and then bring the rest of it in and then stretch out and bring the rest of it in, because they'll use those suction cups to move, and they'll do it quickly as well. But what else you might encounter in your stream is a planarian, and they're a little bit different. They are a type of aquatic worm. They're not actually on our data sheet, but you can always make a note in the comments if you see a planarian. If you remember in biology, we had the fun experiment of having planarians, and these are the type of worms that you can cut in half, and then you've got 2 worms. And they often also use budding as a way to reproduce as well. So you might see a worm coming off of one. So they are very simple in the way that they reproduce, and you'll definitely see those eye spots. They kind of look cross-eyed. What's also fun about flatworms is that they are as basic as it gets. Their mouth and their anus is the same opening. So, fun fact. Okay, and so in the sensitive category, we had the gill snail, which was when you have the top of your hand. Of course, I'm not sure if I'm showing you right or left. I'm using my right hand. But when the opening is to the right, you've got a gilled snail. But when you've got a pouch snail, this is an actual air breather and will take a bubble of oxygen, of air from the surface and take it down. Their opening is going to be to the left. I'm using my left hand. And their shells are much more delicate, and they do not have that little trapdoor operculum. So they are true air breathers with a rudimentary lung, and they're going to be found anywhere where there's a lot of oxygen deprivation. But that doesn't mean they're bad. They're still going to be found pretty much everywhere. So again, every one of these organisms has its place in good quality and poor quality water. Again, diversity is the key. And then the other type of snail that we have that's also an air breather in that tolerant category is the other snail, or sometimes called the ram's horn snail. So they're flat instead of with that spire on top. But these are found in every type of aquatic habitat possible. Okay, quickly, and we'll take a break here in just a moment. We do encourage you to collect your own specimens. Now, the great thing about modern technology is that our cell phones are really good at doing that as well. So if you don't want to take organisms out of their natural environment, you can take a photo and have a photo reference collection. But if you do want to have a reference collection to be able to look at in the future and to help kind of remember some of these identifications, we do recommend using denatured alcohol, which is typically going to be about 95% when you purchase it in any big box hardware store. it will be labeled as denatured alcohol, and you'll dilute it to about a 75 to 80% solution, so you just add a little bit of water. This is the best method for euthanization and long-term storage. It is more expensive than isopropyl alcohol. The last time I bought about a gallon of denatured alcohol, it was about $15, but that will last you a very long time. And the best thing to do in some of the smaller bottles are the green type of denatured alcohol. And so that's actually better because the larger jugs of denatured alcohol you get might have more methanol than ethanol in it, which can be harmful to you. So just take a look at the ingredients. If it's primarily ethanol, that's going to be the best preservation method. But if you decide to use isopropyl alcohol, which rubbing alcohol is something most of us have in our homes anyway, you can do that, but you're going to want to dilute it much more because it is a lot harsher. It's a good euthanizing method, but for long-term storage, you want to have a 40% alcohol, 60% water solution. Isopropyl will bleach out your specimens, and so it's good to buffer that harshness with a drop of glycerin if you happen to have that on hand, or some calcium carbonate, so your antacid for when you have heartburn, just a little tiny crush a little piece of that up and throw that in with your vial, and that will help kind of buffer that solution and make it a little less harsh. So, and of course we know isopropyl is pretty cheap and easy to find anywhere that you go. But also take a look at the type of concentration that you have. And again, you want to have only about a 30 or 40% alcohol solution when doing your long-term preservation. And then finally, hand sanitizer. We do not recommend euthanizing with hand sanitizer. However, if you'd like to use hand sanitizer as a way to display your invertebrates, this can be a kind of a fun way to kind of have it in that 3D type of environment where you can move the vial and you don't have to shake it to see different parts of the organism. However, this is not a good long-term storage method for anything with any kind of meat to it. So definitely not good for crayfish, snails, clams, large cranefly larvae, anything that's kind of squishy and meaty, we don't recommend using it. But it's really good for things like midges. Riffle beetles, very small mayflies. So some of your smaller organisms, this is a great way to display those critters, especially if you have kids and you want to be able to see all sides of that organism. But please euthanize before putting them into that sanitizer because that is a long, slow, painful death. And it will break down over time. Oh, that's another thing. It's really important if you do use hand sanitizer for a long-term solution, long-term preservation, you will have to replace it, but also you need to make sure that you are filling that vial up to the very, very tippy top. You don't want any air in that vial at all, or it will immediately start to break down. So always fill that completely to the very top if you do decide to preserve long-term in hand sanitizer. Okay, now that we've got a quick rundown of macroinvertebrate ID, which I know is probably already left the brains, and that's okay. Again, we do cover this in every level of monitoring workshop that we offer. And of course, any questions that you guys have, please feel free to contact us directly by email or phone, and of course, during the session in the chat as well. So we do have a lot of these kind of fun little cartoons, and this comes from, I believe this is one that I'm not sure if it's actually in that book, but we have a really great resource at level 1, and that is the Stream Keepers Field Guide. And so it has a lot of really great visual information that kind of helps, solidify some of that information that we talk about in monitoring. But, of course, this whole macroinvertebrate session is all about collecting data. Data, however you decide to say it. It's all very important. And of course, over a long period of time, we can find trends within that stream condition. Okay, so those of you who make it through all of these presentations and are willing to take the short quiz in order to qualify for field sampling, will will actually receive equipment from us, and that will include a 3x3 foot kick net, which is essentially about a meter squared, and that is the area of stream that she'll actually sample. And we won't provide the poles that go into that, so you'll want to do that yourself. And I use just PVC pipe that will slide right into the sleeves. And you can also anchor it. There's a sleeve at the bottom of the net that you can put a chain or a weight to kind of keep that net on the stream bottom. We'll also provide you with forceps, and we have two different kinds of forceps for picking bugs, and that your typical kind of hard steel forceps, and then also flexible steel, which is a lot more gentle on the invertebrates, and they're harder to crush. So if you are a teacher and you have a lot of kids that you want to monitor with you, those flexible steel forceps are really great. And then the kids aren't poking each other with the hard pointed ones. We do provide magnifying lenses. But what you'll actually want to provide yourself in your field kit is some kind of sorting pan or tray. And white ice cube trays are the best because you can see the organisms easily. So don't use a blue or cream colored type of ice cube tray because you're just not going to be able to see what you need to see as well. You'll also want to have a squirt bottle of some kind to help refresh the net, pouring water onto the net to keep the bugs from drying out, and that'll also actually kind of wake them up a little bit as well and get them moving so you can see them better. And then in some cases, we may be able to provide a dip net, a long handle dip net. We typically want you to go through level 1 first before providing that because it's a lot more expensive. But if you're monitoring alone, that can be handy as well. So for biological monitoring, we want you to collect 3 net sets within your 300-foot site that you've established. Once you've submitted your site map and discharge, that area that you've chosen to monitor should have hopefully enough habitat to collect 3 nets within a riffle. And that's going to be 3 microhabitats within that 300-foot area. Because even if it's one long riffle that you can get 3 net sets out of, you're still collecting different microhabitats that could have a different assemblage of macroinvertebrates. And we do only want you to really monitor twice a year if you can. So, if you want to do it more, I wouldn't do it much more than that just because you are disturbing the habitat. But late winter, early spring, so right now is the perfect time for those organisms. They're growing, they're getting big, they want to emerge here pretty soon as the weather warms up, and so you can see them better. And then also again in early fall. So September 15th, October 15th, or before all the leaves fall off the trees in the fall, because nobody wants to spend a lot of time picking through leaves. So that's what we recommend. So we have 4 primary habitat types or aquatic faunal regions here in Missouri. But riffles are going to be the type of habitat that we really emphasize that we want you to sample from, because that's where you have the most dissolved oxygen. It's that water tumbling over the rocks, introducing oxygen into the environment, and we're going to find the most sensitive organisms. However, in areas where you may not have a lot of riffles, root mats are going to be also productive habitats. And so this list of riffles, root mats, snags, and non-flow are kind of the priority list, and riffles are at the top of that priority list. But we have 4 aquatic faunal regions. That includes the Big River, Missouri, Mississippi rivers, and then their major tributaries, the lowland region or the boot heel in that Mississippi alluvial valley, the Ozark region, which is a good chunk of the state where you're going to have the most riffle habitats, and then the prairie region where riffles do still exist, but if you struggle with the site that you've decided to monitor, again, we'll talk about these alternative habitats as well. So your Ozark region is going to be primarily riffles and root mats, but again, riffles take the priority, so if you can get 3 of those within your 300-foot site, that's going to be ideal. In prairie and lowland regions, again, there are riffle habitats in many streams. You just kind of have to look for them. So choose a good site that's accessible, that you can get that riffle sample if you can. But root mats are also productive, and then snags and woody debris can also be sampled as well. But the lowest priority are the non-flow and pool areas, so we really don't recommend that, if possible. Okay, again, for review, that's the riffle is the area that's shallow, where we have a drop in the gradient in the stream, so you're actually losing altitude, and then that's where you get the most oxygen introduced into the stream, because it aerates that water and brings in the sensitive organisms, and a lot more food sources as well. So here, this is actually a pretty short area, but we have 3 potential sites that we can sample a riffle in a very small area. And that's 3 different microhabitats that may have totally different assemblages of these bugs. Okay, so in order to sample, pretty simple, place the net in the riffle. You are facing upstream, so you're essentially creating a pocket of the water flowing into that net. And make sure that the bottom of the net is on the stream bottom, and the best way to do that is, if you don't have a weight or a chain or something that's going to hold it down, is to take big rocks if you can and just weigh the bottom of that net down, because we don't want anything to flow underneath that net. And then if you have an area where you might have some large stones right in the area that you want to sample, you can move them aside, and the best way to do that is just to pick them up and rub them into the net, move them aside, and then if you can, remember to put them back when you're done sampling. And then in order to sample, you are just going to use your feet. You can also use, if you want to use a garden rake or anything like that, you can do that as well, but really your feet, your heels are the best tool. And then you just dance and kick, or what we call the benthic boogie, and you want to get as deep as you can in that substrate, and 3 to 6 inches at minimum, because we do have a lot of organisms that do burrow themselves pretty deep down into the substrate. And then really you just go until that about meter squared or 3 by 3 foot square area in front of your net is thoroughly disturbed. And again, that's why we only ask you to sample twice a year, because we are disturbing the environment. So here's an example, and the net is placed facing upstream so that it's creating a pocket, and April's putting rocks down to kind of weigh that in and allow the water to flow in. And then doing the benthic boogie, and I like the twist. Twist those feet and those legs, and that gets you down pretty deep. Okay, and then once you've thoroughly disturbed the habitat, and we used to say 3 minutes when I was trained a long time ago. 3 minutes is actually a really long time when you're really getting into it. So just until that substrate is thoroughly disturbed. But when you pick up the net, just be very careful not to allow the water to go over the top. Where Jenna there is holding the poles, we don't want the water to go over the top there. And the best way to do this is to pick it up by the bottom and not by the poles, because sometimes that water is going to rush out if you pick it up by the poles. So definitely pick it up by the bottom of the net and very carefully let that water drain. And then you'll move to land to pick and sort those invertebrates. And the thing that you want to do as soon as you're ready to pick is make note of the time, because you're actually going to track the time it takes to pick those invertebrates. I froze up here. Okay. All right, if you decide that, or if you find that at your adopted site, you don't have enough riffle to get 3 sets out of that in your 300-foot site, you can also sample a root mat. And a root mat is simply just the area where you have vegetation hanging down and nice live roots from a stream or from a shrub or a tree hanging into the stream. This is a really great habitat for dragonflies because they're site predators, but they can hide themselves pretty easily. And then in our prairie streams, we can actually find a lot of those stoneflies and caddisflies and mayflies in that root mat habitat. But definitely look for live roots, those soft fibrous roots. And here's another example. Yeah, especially in our prairie streams, this is a very productive habitat. But if you do sample a root mat, try to sample that root mat again the next time you go out. We want as consistent as the area of where you sample as possible every time you go out. Okay, so using a kick net, again, you're facing the net upstream along the side of the bank, and this is really good. It's good to have a buddy. So hopefully you all will definitely have a buddy when you go out to sample. And then you're simply going to agitate those roots by kicking and swirling the water into the net. Now, it's always good to make a noise when you go into a root mat, because if you have an undercut bank, you might encounter things like snapping turtles and possibly water snakes, depending on where you are. So making noise to kind of scare those critters away is recommended if you do end up sampling a root mat. And then again, just carefully removing that net without allowing the water to go over the top or the sides of the net, and picking it up by the bottom. Okay, now if you're really struggling and you don't have enough riffle and maybe even not enough root mat in your 300-foot adopted site, you can sample woody debris or snags. So snags is essentially just a short word for woody debris that's fallen into the stream. Anything that's decomposing and has a lot of kind of spongy type material to it is going to be a good habitat for a lot of things, especially like medges. I found stoneflies in some of that habitat, that wood that's kind of been there for a while. And depending on what size of the piece of wood that you're sampling, you can take smaller pieces and use a scrub brush to just scrub anything that might be living on it into the net. And then, or if you're looking at a larger tree limb, then of course you're going to be facing your net upstream and then rubbing and scrubbing into the net. And if you do end up doing woody debris, 3 to 5 snags can equal 1 net set. So use your best judgment if you do end up using woody debris, and 3 to 5 smaller pieces of wood is 1 net set, or perhaps 1 large piece of wood could be a net set as well. Okay, we don't recommend doing non-flow areas, but if you do, this does require the dip net, the long-handled dip net. Again, we don't recommend sampling non-flow because you're going to find a lot of organisms perhaps that aren't part of our data sheet. Most of those would be the Hemiptera, those true bugs that are either living on the surface or up and down the water column and not necessarily in the benthic zone. But if you do, and you have a long-handled net, simply hold that D-frame net downstream of where you're standing. So you're, again, going to be standing upstream to disturb that substrate. But give it a moment. When you're digging into a soft substrate in a non-flow area, you're going to get a lot of sand and sediment. So give it a second and let that organic material flow up after you dig with your feet, and then lightly sweep back and forth through the water to get that organic material. But again, riffles are going to be ideal for sampling. And then just repeat, rinse and repeat. Okay, it's important to remember that when you start sampling, that you start in a downstream section of your 300-foot site and move your way upstream. Otherwise, you might be collecting the same invertebrates. If you start upstream and go downstream, you've already disturbed the habitat. That really kind of messes with the quality of the data. So always move from downstream to upstream. And when you are out there doing all of your monitoring, bugs is probably the best thing to do first, because if you're out there doing discharge or even some of your site selection, or later on when you're doing visual survey and chemistry, you're going to be disturbing the habitat. So we definitely recommend doing the macroinvertebrate sampling first. And then sample those same habitats each time you go out and monitor, if possible. Okay, so we've collected our sample. How do we analyze what we've got? But we want to make sure that you understand that recording the time that you spend picking off the net is really important. And not identifying, but just getting the invertebrates off the net. Okay? And then you'll stop your time after you're ready to start identifying. And then you count your invertebrates and record on the data sheet. And a lot of questions about, well, how long do we pick? Well, that depends. It depends on how busy your net is. And perhaps if you don't have a whole lot of organisms, you might pick it clean pretty quickly. However, if you have a net that's teeming with organisms and perhaps a lot of the same types of organisms, the best thing to do in that situation is, of course, you're going to pick whatever's moving first. And then when you realize you've got 100 scuds in your tray, maybe look for something else. And that will still provide a nice proportion of what's actually in your sample, even if you don't end up getting all of the organisms off of your net, especially when you have a lot of the same kind. But we don't estimate, we count everything that we pick. Okay, so here's your data sheet. Just like all the other data sheets, you're going to have the same header information. The only thing that's going to change depending on your sample is the date and the time and the water temperature, but we will provide you with a thermometer as well. And always be consistent about that site location description, because we need to be able to find that on a map. And then, of course, your date, including the year, your military time, your time in military time, rainfall as best as you can get with the information that we can provide. We have some of those resources on our website. And then, of course, your participants and your trained data submitter, which is you. Now, something that often gets overlooked, because this is a pretty small part of the data sheet, is making sure to identify that habitat type, okay? Because we can't assume. Laura will never assume what type of habitat. So always make sure to include the type of habitat and the type of net that you used. Okay, this is important information. And then, of course, you are tracking your time that you spent picking, not identifying, but just picking and getting the invertebrates off the net, and the number of people. And that gives you people minutes and the amount of effort that you put into that picking effort. Right? And then for each net set, you're going to count all the organisms. You're going to identify and count and provide the accurate number for each type of organism. Okay, and you're going to do it 3 times, again, moving in an upstream direction. So reporting actual numbers, we're not doing any estimates. I know sometimes folks will do hash marks on their data sheet in the field, but if you do that, which we don't recommend, we just have, if you can just count it in your mind, that's great. If you do end up using hash marks, make sure to add them up. We can't, if you do your data online, you're not going to be able to have hash marks. So just make sure that if you're just field marking, make sure to add those numbers up. And we're not just doing check marks, we are counting the actual number of specimens. Okay. It's also really important to remember that we do have to have all 3 net sets in order to process your data. Life happens, weather happens, sometimes streams start to rise quickly and you may not be able to finish. In that case, we would just recommend going back to get that last net set when it's safe to do so. But we do need all 3 net sets to calculate the water quality rating. And again, it's because you have those 3 different microhabitats, and you'll find different things in each net set, even if those riffles or those habitats are close to each other. Again, like every data sheet, we have the instructions on the back, so that's a good reference point if you're not sure. Okay, so I mentioned that we're not doing check marks, that we are actually having to put those actual numbers down, and I'll tell you why. And if you need to, you should have the ability to zoom in. And let's see, I think I can do this as well. So I will zoom in a little bit so you guys can kind of see this a little bit better. So we have our header information. So we have a sample that we went out and took on Turkey Creek at the same site and that we've been doing for years and years. And we found that when we were out there, we collected our 3 net sets. Oh, this is not the right one. Let's see. I think I may have accidentally gone 1 step too far. Well, there we go. Okay, that's the one. My apologies. So let me zoom in again. Okay, so we went out to Turkey Creek and we collected our 3 net sets in our riffle using a kick net and recorded our time picking. And so for every organism that you find in any of the 3 net sets that you collect, you're going to circle that type present. So for example, a riffle beetle was only found in that first net set, but we are still circling that 3 because it's present, okay? But there's a reason why we're not just doing check marks. Yeah, after we did our 3 net sets and we went through, we found that we had a water quality rating of 25. So that is pretty standard. That's in that excellent range. So not much to worry about there. Although I think I'm still on the wrong slide. Yeah. Okay, the first time we went out, we had the water quality rating of 25. So everything's good and dandy. The second time we went out, and here we go again. We recorded all the same types of organisms that we saw before. However, we had a little bit of a population change. And so we found that even though we had some of those sensitive organisms and we circled all those types present, we found that we had a lot more quantity of the more tolerant organisms. So we still had a water quality rating of 25, but something happened to the population to where we are seeing a lot more of the tolerant organisms and a lot fewer of the sensitive. So even though we had the same water quality rating, something changed. So that's why it's important to always record the actual numbers, because you may end up with the same water quality rating, but you're seeing a shift in population. And this data is important over a long period of time because that stream condition is going to be stable in a stable environment, but can change quickly based on watershed land uses. So I'll always record those numbers. Okay, so a lot of things can affect your water quality rating. The type of substrate you have available. Some streams may have really great water quality, but perhaps it's full of bedrock and just may not have the type of habitat that would be conducive. So there are streams that they're wonderful, they've got good watershed practices, but there's just not a lot of those interstitial spaces or places for the invertebrates to live. That's just a lack of habitat. It's not a lack of water quality. The type of discharge, which means immediately after a large rain, you want to wait until things have settled down a little bit, because those invertebrates will release from the rocks that they're attached to and drift downstream. So after a large rain, you want to wait until you're kind of back at what we call base flow in order to do your invertebrate sampling. Water temperature can have an impact. The dead of summer, July and August, boy, that water's hot. It's not holding as much oxygen, and perhaps if it's in the summertime, most of your invertebrates, at least the insects, have already emerged, and so you're not going to find the same diversity that you would find in spring and fall. Right? Stream size can have an impact as well. Water chemistry, again, oxygen is a big one. So if it's really hot outside, you're not getting as much oxygen, you may not see the diversity. Oh, yep.
I'm going to pop in with a couple of questions back on the data sheet for you real quick before we move on.
Sure. Sorry about that.
Nope, that's all right. We're just trying to play catch up on a little bit of it too, and didn't want to go too far out. Can you explain a little bit more on the objective of measuring these numbers? Is it more the type of macro or the variety? And explain a little bit more on the circling of the types present.
Okay, sure. That's a really good question. We do get that a lot. So, okay. Yeah, diversity is always going to be the key. And even if you have tolerant organisms in your net set, always count them because they all have a place. So diversity is the most important thing. Because you're doing 3 net sets, that's why you are circling what you find in any one of those net sets, okay? So you had hellgrammites at each net set, that's great. So you're gonna circle that 3. You only had a riffle beetle in that first net set, but it's still there. And so you're still gonna circle that 3 because this designated number, the 3 for the sensitive organisms is the higher ranking, 2 for your somewhat tolerant, and then 1 for your tolerant. Okay, and so when you add all of these numbers up after all 3 net sets, that's going to add up to your water quality rating. Again, your sensitive organisms have a higher number assigned to them because they, I don't want to say that they're more valuable, but they do increase the quality of your stream. So does that help? You can see there was a fish fly in just one of those net sets, but we still circle that 2 because it was present in a sampling event. Is that good? Okay. All right, no, thank you, I appreciate that. Sometimes I feel like I don't always explain that very well, so. Okay, were there any questions before I move on to more factors that affect water quality rating?
No, I think you're good.
Okay, thanks. All right, so I think I mentioned a little bit about water chemistry and I talked about oxygen. That's your primary parameter that these organisms need the most in order to survive. So, you know, if anything has taken the oxygen out of that stream, that's going to affect your water quality rating. Physical factors such as channelizing the stream, removing the riparian corridor, which is simply just all of the vegetation along the stream, can have a huge negative impact on your water quality rating. So ideally a stream should have that sinuosity, that it's kind of meandering type of shape to it, with plenty of vegetation along the stream. Because again, root mats are a very productive habitat, and that's where a lot of things live. So take those away, and that's going to immediately affect your water quality rating. And then the fact that we also are identifying these organisms in the field mostly to the order level. If we wanted to go into genus and species, that would require a microscope and extensive keys, dichotomous keys in order to identify them. And we can get there, we can do that, but for you guys, you know, being out in the field, we don't expect that. So identifying to order is going to be the easiest thing to do in the field. It's still fun and it's still absolutely valuable information. Okay, so we have some scenarios here. I always use, for some of you who might be here in the central region, I always use Hinkson Creek and Flat Branch Creek as my examples. But we're just going to have a few scenarios and just kind of give, or just kind of speculate what we think might be happening based on what we find in our macroinvertebrate samples. So the first time I go out to Hinkson Creek here in Columbia, and my general observation after collecting my 3 net sets is that I have high density, so a lot of invertebrates, high diversity, a little bit of everything, and a lot of that sensitive taxa. So what can be our general observation or general conclusion from that particular sampling event? And go ahead and drop that in the chat. Just general observation, high density, high diversity. Yeah, that's pretty simple. No problem. Things are looking good. So I decided to go upstream in the watershed and go to Flat Branch Creek. And it was a beautiful day. My general observation though, and if you're from Columbia, you might get this one pretty quickly, but my density was pretty low. I didn't have a whole lot of invertebrates, but I had really high diversity. So I had like one or two of a little bit of everything, but just not a whole lot of each kind. What might be a conclusion you could draw from that? And I did allude to this earlier in water quality factor, things that affect the water quality. And I'll let you guys kind of think on that for a second. So yeah, I had a little bit of everything, just not a lot of everything. Yeah, perhaps there could have been a rain event recently that might have put some of those invertebrates downstream. But perhaps it's just simply poor habitat. And for those of you who are from Columbia, you notice that Flat Branch Creek is mostly bedrock. And traditionally, it can have a pretty good diversity, but just not a lot of quantity, because it just doesn't have those interstitial spaces or those little nooks and crannies that those invertebrates need. Okay, so next I go back to Hinkson Creek, the site that I was at before, but something changed. I have a whole lot of bugs, but only a few different types. So I got a ton of midges, I got a ton of scuds and sow bugs, but nothing else. So I had 100 of scuds, 100 of sow bugs, and a whole bunch of midges, but just I didn't have that diversity. What might have happened from the point where we had good water quality and there was no problem up until now? Yeah, there could be some kind of pollution. We have really high density, just not a lot of diversity. Okay, so yeah, there's some kind of pollution, but what kind? And this would actually be a really good indication that it could be organic pollution or nutrients being put into the system. A lot of nitrogen and phosphorus that could come from pet waste, from fertilizers on our lawns. And that's something urban streams definitely have, that's an impact that urban streams have. So we could have a lot of algae growth because of those nutrients. And I think I mentioned that the sow bugs and the scuds really love that organic material, and so they will thrive in this type of situation. So this is a situation where you might find a high density of certain somewhat tolerant and tolerant organisms, but not a lot of diversity. Okay, so last scenario, we went back to Flat Branch, the same site that we did. We had good diversity, just not a lot of density because of that habitat issue. But this time we went back and there was nothing. There was nothing there. No diversity, not really any bugs at all. But the water was clear as a bell. It looked beautiful. What do you think might have happened? Yeah, definitely some kind of pollution. Yikes, yes. And this has actually happened. This is a real scenario. Toxic pollution of some kind. And yes, the draining of a pool, types of metals and pesticides, so some kind of inorganic type of pollution. And this actually happened on Flat Branch Creek several years ago when our O'Reilly car parts place caught fire, and a lot of the fire retardants and the treatment for tending to that fire polluted the stream. So these are absolutely very likely scenarios that can happen, especially in an urban stream. Okay, we're almost to the end of the biological chapter, but we also want to make sure that we are taking into consideration endangered species. Niagara darters in particular, they do spawn in riffles, and so of course we want you to sample riffles. But if you are in the type of habitat where, or the type of stream or watershed where a Niagara darter might inhabit, we don't want you to sample between March 15th and June 15th because that is their prime spawning period. And for a little more information, so these are the particular counties and watersheds where you might find the Niagara darter. So if you are sampling in any of these areas, We want you to avoid doing macroinvertebrate sampling between March 15th and June 15th. So just to take into consideration that we want to preserve that endangered species. And the highest remaining populations are in the Niagara River, the Little Niagara, Tavern Creek. I believe there's several others, but if you can identify the county that you might be sampling in, these are the areas to avoid between March 15th and June 15th. Oh, I see the question, how large are the darters? They're actually a little bit larger than your typical rainbow or orange-throated darter. So I've only seen one or two, but they were actually about 4 inches long. So they're just a little bit larger than your more common darters. Okay, also to consider are those aquatic nuisance species or those invasive species that we don't want to spread. And that can include the zebra mussel, the Chinese mystery snail, rusty crayfish, which thankfully we don't have yet in Missouri, but something we're keeping an eye out for. And then hydrilla, which is an invasive plant where I believe most of the populations that we are monitoring are in Southwest Missouri near Springfield and around Kansas City on the eastern side in Johnson County. These plants are extremely invasive and will take over an entire waterway. So always something to keep in mind that we don't want to spread species that don't belong in those waterways. So the best way to do that, if you're planning to sample multiple streams in a short period of time, make sure that you are treating your equipment, and that includes your net, and that includes your boots, anything that's going to go into the water. Vinegar, chlorine, actually air drying is simply the best, but if you're going within a short period of time, freezing or salt is also effective. And this information, I think we have this on the Stream Team website as well.
This chapter is just going to cover some mapping and online tools that we have, so you guys can get a little bit more familiar with it and kind of feel accustomed to some of the tools that we have with our resources. So we're going to talk about, again, kind of watersheds and mapping your watershed, if that's something you're interested in, as well as topographic maps and some online tools that we have available. So the importance of watershed mapping. We talked a little bit about this last time, but again, it is helpful to know what's going on in your watershed to really understand your water quality within your stream, right? We talked about how the watershed is topographically defined and that rainwater runs across all of that land area before it enters the stream systems. So what's going on in your watershed could have an impact on your stream. So mapping your watershed could be helpful for you or your community to better understand areas where improvements could be taken or best management practices could be put to use. Sorry, here we go. Okay, so again, we kind of also talked about last time how there are natural and kind of land development that can occur in a watershed. With more natural watersheds, you get a lot more water that percolates through the soils. But when we develop on a lot of land, again, it causes a lot of that water to run off easier and then enter into our stream systems. So understanding what's going on in our watershed could help us interpret what's going on with our water and streams in regards to water quality. So if you are interested in, again, forming a map to help interpret your watershed and the health of your stream, think about choosing a manageable size. So again, think about your main stream and all of the tributaries that run into your stream. For some of you guys, this may be small, but some of you guys may have a larger site with more tributaries as well. So just think about that when you're mapping out your site. And again, when mapping out your site for a watershed, it is defined topographically. So knowing the topography of your region can also help determine the boundaries for your watershed. So that is why we still look at topographic maps. Again, topographic maps are relevant for, in my eyes, hiking. You know, how hard is my hike going to be? Because they illustrate relief, right? So they are maps that show elevation. When you're looking at a topo map, it is helpful, like most other maps, to look at the scale to look at the key to learn A, the scale, and B, some of the symbols that are on your map that can help give you more information. So this is an example of some of the keys you might find. Again, you can see here that a lot of the lines on a topo map demonstrate elevation, and your topo map could have information in regards to your river, like a perennial stream or different types of streams. So look at your symbols when you're looking at your topographic map, if that's something that you want to use when looking at what's going on in your watershed for assessing your site. So here's an example of a topographic map. Again, you can see the contour lines. So these lines help illustrate a change in elevation. So the more lines you have closer together, the more steep your elevation is getting. When looking at a topo map, again, you're looking at how hard your hike is going to be, right? It is a one-dimensional thing that shows something that is actually kind of got elevation to it. So thinking about your hand versus a palm explains topo maps. Again, when looking at topo maps, you'll find kind of lines that contribute to a circle. And you need to take note on whether those lines are forming a valley, so whether they're going down in elevation, or whether they're forming a ridge or going up in elevation. This can also help determine which direction that stream that is on that feature will go. So if you look here, the elevation outside is starting at 800, and the closer you go in is going to 650. So the elevation is going down in this valley. So the stream is flowing down in this direction. So if you're thinking upstream versus downstream, this V illustrates the upstream direction. For a ridge, again, elevation is high, and then it's decreasing as you get away from this center circle. So that if there was a river, it would be going down off of this elevation, right? Because all of our water bodies are flowing down topographically. So when you are looking at a topo map, please be sure, again, you are looking for features such as water bodies, and you're looking at, again, developed areas versus maybe some more natural areas like forests. So those are things to be cognizant of when you're looking at a map. Also roads, again, another sign of development. So this is what I'm talking about when I'm talking about the circles can help illustrate some of the higher regions. And again, these lines are illustrating how hard your hike is going to be. And they interpret to real life function. So this is what you're seeing on a map, but this is what you would see when you go out for your hike. And this is the water body that's forming here. So for some streams, a topographic map could be helpful for figuring out your watersheds. If you're looking at this map, you may be like, oh my gosh, this is a lot of stuff going on here. But we have some helpers that can help you map out your site. So for starters, you'd want to draw a line of the river, or sorry, the stream and all the tributaries running into it. So that'll help you pinpoint, again, what you're looking at. And then you would look at all the high points or these circles illustrating the high points, and you draw an X on all the circled high points around your water system. So then to finish off your topographic map, you would connect the dots. And if you look here, this is your watershed. So if you have a smaller stream, topo maps are a great way, again, to look at that direct watershed and to look at what potentially is influencing your watershed. If you like topo maps, they are available on the USGS website, but we also have some topo layers on our resources that we're going to show you that, again, you can use for your own site and to pinpoint a specific watershed. So we're going to go over some of our online and mapping tools. Again, these are all kind of geared towards mapping or getting to know your watershed better. So we recommend using the Ag site with Missouri EDU. What you can do, again, it's super easy to search your water body, and then you can scroll through and click the site that you're likely going to monitor. And then if you see down here, it can generate a report for you with some additional information on your site. So you can see here, this is the site location in this red, and this green is outlining a watershed for that site. And that site area, the report will give you information on demographics near your site, hydrological summary, some soil information, and environmental concerns. So if those interest you, this is a great website for you. How's My Waterway is also a great tool, particularly if you're motivated by the way you use a watershed. So if you like to go swimming in it or you like to fish in it, this is a great resource. Again, this is a EPA website, and it illustrates the watershed with this dashed out line. And it also has more details of what we know about the conditions of the stream. So you can see here with this green, we know this green part is good water quality. It will also show impaired water quality. And this purple triangle here will show conditions unknown. So if we don't know the conditions, that may be a place where we need more information, and more information could be helpful potentially. So the Stream Team website has some online tools that are great to use as well. This is what the Stream Team website looks like. If you are interested in any of the volunteer water quality resources, you want to hit this water quality tab, and they're all available to you. If you want to order equipment, that's under reporting forms, which is right here on the website. Also, activity reports are helpful to submit to us anytime you do a Stream Team activity. We also have online data submission, again, through the reporting forms, I believe. And we have a calendar available for any Stream Team activity that is available and open to the public. And this is where the interactive map is located as well. So the interactive map is useful if you're looking to do any Stream Team activity, or if you're looking to pick up a monitoring site, or if you're looking for Stream Team data. This is where you can find the data available. So if you're using this site, you can easily, again, search your water body and then click on the stream with your water body, and you'll hit zoom to that water body feature. Then you can scroll and drag across the water body until you find the area you're potentially looking to monitor. You can also see all of the icons for spots that were monitored, and you can click on it to see if there's any data. We also have multiple features within our mapping function. So you can see here, you can select what type of imagery you would like present. So if you like the topo maps, you could try a topo map layer. But a lot of folks use the imagery layer. This is a great layer to see again. Okay, look here, this area is forested along the stream bank. That's good because the water can then, percolate through that water in that area. It's not developed all the way to the stream. So you can learn more information by selecting different layers. And that's, again, all these features are located at the bottom of the Stream Team interactive map. You can also select the measuring tool, which is really helpful for outlining how far you are away from a permanent landmark feature. So when you're describing your site, you can use this mapping tool to help you discover, oh, I went this far upstream, so this is where my site is. Again, there's also drawing tools located at the bottom here, and these can help you identify a marker for your site, which is important for submitting your site map. There's also features in which you can add text to the map, which is great because you will have to identify to me your name, the site number, particularly if you're adding more than one, the name of the creek, county, and that physical description of your site. You can also do things like print the site if that is helpful to you using the Stream Team map. So again, when you're looking at your site, there's two different icons. The Missouri icon is an indicator of any Stream Team event that has occurred. So if you're interested in litter pickups, this is a great icon to search through. But if you're interested in data, the beaker icon is the one to click on to learn more data. So again, the Stream Team icon, it has that physical description, so you can easily find that site when you are looking for it. So here's the description of the volunteer water quality monitoring site. If you're inheriting a site, this is a screenshot that you might want to submit to us for your map. It has something called the site IDX, and it has a description of the site on it. So it's a great feature for me to quickly add your name to that site so you can submit data for it. So if you're inheriting a site, this is a screenshot you would want to take to submit to us. If you're wanting to learn more data on this site, you can see right here it says water quality data, and you would select the link that says more information. That will take you to this page where you can select different data sheet types that you might want to look at. So you can select water chemistry, and it will pull up the data collected on the water chemistry for that site. So you can see from this site, it was last sampled in 2005. So this would be a great example of a site to pick up to continue some data tracking on this site. Also, if you want a usable format of the data, like an Excel form, you can contact me and I can send that out to you, and it will have all the different data types on it. So again, when you are establishing a new monitoring site, you're going to want to send that map to streamteam@dnr.mo.gov. When you submit your new site, you want the map to have your name, county, site number, particularly if you have more than one, creek name, and a physical site description of your site. If you're inheriting a site, again, try to get that screenshot of an icon so I can easily add that site to your name. So have that icon include the site IDX if possible. So again, here's a great example of a good map. I can read the roads, the creek name, and they have indicated where they are monitoring on their map, as well as provided all the information that is needed. We talked a little bit last time about how when measuring discharge, you can use a gauging station instead of doing the discharge monitoring procedures that we have. This is only applicable if the site is within half a mile of your site and there's no inputs or outputs. But the USGS has multiple gauges throughout the state. You can see here that it will pull up as a Missouri map, and then you can search per county and then select the gauges near your water body if there are any gauges. So this is what it would look like. And you would, again, select your county and then zoom into that feature to find the gauges. If there is a gauge within half a mile of your site and there's no inputs or outputs, you would, again, look at the gauge number. This is what you would record in your submission. And you can see here we've got information like precipitation, discharge, and gauge height. You would want to select discharge, and then you want to view it in a table format. And then you'd want to select the begin and end date for the date that you went out and sampled. You would then look up the time that you went out and sampled and then record the CFS or the flow on the gauge for the time that you were out conducting your other VWQM data. So again, we can't say this enough. Check to see that the gauge is close to your site. If you're confused on whether or not the gauge is close enough, you can contact us and ask us to help verify for you. But again, we're talking half a mile within your site with no stream inputs or outputs between your site and the gauging station. So another thing to think about when you guys go out and sample is please submit an activity report every time you go out and sample. Activity reports help illustrate how many hours Missouri citizens are putting into this program and helps validate the infrastructure for keeping this program interstate and helping improve our water resources. There's also benefit to you if you submit this activity report. You can request incentive items, which could be anything such as t-shirts or koozies, pencils. We've got a lot of stuff that you can request as swag or give away as swag. And we also have a prize drawing every year, which can be a larger item. I think historically it's been stuff like canoes or kayaks. So there are benefits to submitting an activity report, and we appreciate it if you do. All right, so we're gonna do a quick poll. Again, if you are an operator, please, and you're looking to get credit, please submit your name and certification number in the chat so we can get that credit for you as well. So if you're a wastewater or drinking water operator, please be sure to submit your name and certification number if possible. For the rest of you guys, go ahead and take the poll. Sorry, we're over a little bit, but we will stay on if you guys have questions. I see a question in this chat that says, how do you know if a site shown is available? So that's a great question because it's not easy. But in general, let's just say if it's been more than 5 years, you could probably take over that site easily. If there hasn't been data submitted in 3 years, you can contact us just to double check if it's open or not, and we can get back with you. But that's the best way you can go about seeing if a site's available. If you're not sure, just contact us. We have local people out by the stream teams that can double check on that. So that's a great question. There are no more quizzes. Okay, so after this training, first of all, I want to thank you all for attending our trainings. It means a lot to us. But if you are looking to collect water quality data, the next steps are taking a pre-test, which just verifies that you've taken this class or you attended, or it's pretty much just a verification system. You could take it as many times as possible, and it's open note, so again, it should be passable, you know. And then we will you'll send us like a survey of when you would be open to receiving field training and where you would like your field training to take place. And then we will follow up with you to train you guys in the field. We know that we're giving you guys a lot of information, so the field training is just to help you guys kind of be more secure when you go out to the field for your first monitoring trips, and then we'll let you on your way for the discharge and biological monitoring. So those are some of the next steps after this, and we will send a follow-up email again with those steps, so you guys can take those if you want to. All right, so I'm gonna go over the poll. So, oh, I gotta extend it out first so I can read it. So how is a watershed boundaries defined? So they are defined topographically. So using the contour lines will help determine where the high points are, so you can see the boundaries of your watershed. So the contour lines help form the boundaries. What do the contour lines indicate? They indicate elevation, so how hard your hike will be, pretty much. Where can you find the interactive map? That is on the Stream Team website. And when can you use the USGS gauging station? It's when it's within 0.5 miles of your site with no inputs or outputs. So you guys did pretty good on the polls. Again, thank you so much for staying on and taking our training. We really do hope to see you guys in the field, and I hope to see your data personally. If you have any questions, I'm going to stay on for a bit and I can answer them in the chat. We do not need a drawn contour map. We just need a map that I can figure out where your site is. So the drawing of the contour map is more for your purposes, potentially for knowing what's going on in your watershed. So say you go out and you get a water quality rating, or when we get to level 1 and you get a water chemistry rating that might be off, you might want to start thinking about, where could this reading be coming from in my watershed? So that's why we talk about contour maps, is just because they help define what a watershed is and what potential impacts there could be in your local watershed, in your local stream system. Oh, we don't need one. You can provide it if you want. You know, it's all good information, but it is not needed. So for getting to the next level, so we'll start with how to complete this level again, because it's a bit of a transition for us because we used to just do 1 day full event, and now we're trying to do online and then a field portion. So I'm just gonna repeat it for myself, maybe. But for completing the intro training, you have to complete the online portion, A and B. Then you have to take a pre-test that just helps verify us that you attended both, because it's kind of hard for us to track because some people could lose internet or whatnot. So the pre-test is just to verify that you took it. You can retake that as many times as you want, and it's open book using all of our resources. And then you can work with us for when you feel comfortable for a field training to occur. Once you complete the field training, basically we need a site attached to your name. We need a discharge data sheet and a macroinvertebrate data sheet, and you're eligible for level 1 training. Level 1 training is beneficial because we cover the water chemistry and the physical features of your stream, as well as some more kind of help and development on the macroinvertebrates. So for the next level, for level 1, you would need to create a site or add yourself onto a site and then submit a discharge and a macroinvertebrate. In order to do that, you'd have to attend a field training. We will send out the pre-test and the application to attend the field training after this training. So you will get an email on it, but you can also contact us. We have those links, and those links are available on our website, I
I
believe.
put the link in the chat
Okay, the
too.
link's in the chat, and it's also on our website under the water quality monitoring tab, and you can select intro training, and then you can find the pre-test and stuff online.
And I'll also send it out with a follow-up email with the video link as well.
Perfect. Were there any more questions that I missed in the chat?
Hey, Laura, I just wanted to address, let's see, let me go back up here. For Cassandra, who is part of an established stream team, moving to the next level, volunteer water quality monitoring is independent of what your team activity does. So it's tied to you personally as a stream team member. As Laura mentioned, taking the quiz to qualify for the field training, to be certified to collect data, is for you, regardless of what the rest of your team does. And of course, sites that you choose are also tied to you as an individual and not to the team. So this kind of functions a little bit differently than some other stream team activities. So I hope that answers that question.
Thank you, Amy. And Deborah, to answer your question, we send out all of our reminders and all of our announcements through the stream team constant contact. So we're still learning with going virtual. So our level 1s are typically in the fall. And so if we have level 1s in the fall, we'll announce it through the stream team email, and it'll be on our website.
Oh, and Cassandra, for the field training, nope, you don't have to have someone else join you, because we'll be there to demonstrate those methods. I mean, you can if you want to, but if they didn't come through the program, they won't be certified until they go through Part A, Part B, the quiz, and the field training as well.
Yeah, I feel like our training has a lot of information, so really seeing it in person is helpful, especially when you're going to go out on your own and start discovering this own, your own site in person. So that's why at this time, we're still having the field portion for in-person training.
And yes, one of the stream team staff members will coordinate the field training.
All right. If you have any questions, I'm someone who thinks of a question 2 days later. You can always email us. All of our emails and contacts are available on the Stream Team website. So please feel free to reach out to us. That's what we do. So we'd be happy to answer any of your questions or assist in any way that we possibly can.